Good afternoon, everybody. We're going to try to start on time because we have a really interesting session here. In addition to a talk by our invited speaker, Trevor Pugh, we're also going to have a panel discussion. So we're really going to be able to understand how ctDNA might be changing how we practice medicine, especially when we're talking about precision medicine. So to start off, Trevor is a cancer genomics researcher, and he's a board-certified molecular geneticist. He's really been at the forefront of precision medicine. He holds the Canadian chair in translational genomics, and he uses genomic sequencing to understand the causes of cancer, how to guide treatment for patients, and detect early disease using blood tests. He is appointed a professor at University of Toronto Department of Medical Biophysics. He's a senior scientist at the Princess Margaret Cancer Centre and a senior investigator at the Ontario Institute for Cancer Research. Very pertinent to the work that he's going to talk about today, he directs the OICR Genomics Program and the Princess Margaret Genomics Centers, which are the genomics cores that specialize in single-cell, cell-free DNA and clinical accredited whole genome sequencing. He has contributed to multiple and important large-scale genomics and data sharing programs such as the NCI Target, AACR-GENIE, and more recently, and very pertinent to our own oncology axis, the Terry Fox Marathon of Hope Cancer Centers Network. He also leads the CHUM's consortium. He smartly uses the ADAM software for his projects, which makes me very happy. And the CHUM consortium has really been to initiate the use of cell-free DNA for all people with inherited predispositions for cancer. So without further ado, I invite you to give your talk.
Thanks, Anne-Marie, for that very nice introduction, and really for the opportunity to come visit the CHUM today. I've actually never been here, despite collaborating with so many of you over the years. Speaking of collaborations, I'm going to talk, rather than one project in great depth, I'm going to talk more broadly actually about 3 projects where we've used the exact same technology, whole genome sequencing, to understand detection of cancer, management of cancer, and monitoring of cancer over time. It's sort of a map of what I'm going to talk about. I'd argue cancer genomes are always informative, regardless of where in the cancer patient journey an individual may be. Relevant at cancer predisposition, trying to understand your risk. Certainly relevant at diagnostics, trying to understand what type of cancer, what are the drivers, what are the treatment options. And then upon monitoring, but also eventual relapse, precision medicine sort of grew up in this world of clinical trial matching, sequencing genomes, then using gene panels, now using whole genome sequencing to assign patients to trials. And in translational research, we love to draw curves like this. We pretend that we know the tumor burden at every point in time, but in reality, these tests are piecemeal and they're at single time points. So we're actually in the dark most of the time. You might get one test as a risk predictor, you might get a test at diagnosis, and you may get maybe a couple of tests as you go through clinical trials. But we actually aren't able to paint those elegant curves over time. And really what my talk is all about is using cell-free DNA as a spotlight to trace out those curves, being able to do more frequent testing, but also to be able to not only find cancer early, but to really pull out the entire cancer genome, even upon cancer relapse, to assign patients to clinical trials and have new precision medicine strategies based on the cell-free DNA alone. What is cell-free DNA? All of our blood contains small snippets of DNA that are free-floating in our bloodstream, and actually we've inherited a huge amount of technology from non-invasive prenatal testing. This is now a test that virtually every pregnant person will have. You get a shallow whole genome looking for aneuploidy. The challenge here is fetuses are relatively large, tumors are often quite a bit smaller than them, but the concepts are exactly the same. Sequence the blood and try to find fragments coming from a certain organ, person, and try to not only say where is the signal coming from, but what are the biological mechanisms behind it. Cell-free DNA sequencing is often described as needle in a haystack, but I would actually argue that haystack is actually full of millions of needles. It's not just a single needle. There's many opportunities to find cancer-derived DNA in the bloodstream. You just need the right magnet to pull all those needles out. So each tumor is shedding in billions of cell-free DNA fragments per hour. So our challenge and tool that we're using for this is cell-free DNA sequencing. We sequence every fragment circulating in a blood sample, and then we use computational techniques to analyze and differentiate cancer fragments that are derived from cancer cells versus fragments that are derived from all the other cells in the body. So how do we do this? I'm going to circle back to this idea at the end, but a single cell-free DNA fragment actually packs a huge amount of information into it. Certainly the sequence is important for understanding mutations, but there's other aspects of that fragment that allow you to tell the difference between a cancer-derived fragment and a normal fragment. The end motifs tell you how that fragment was digested or fragmented. Certainly a lot of technologies to measure methylation marks in addition to mutations. My lab has special interest in T cell receptor and B cell receptor sequences, tells you about immune responses. Where the fragmentation happens, give you interest of information into the open chromatin state, so the cell of origin of the fragment, and actually the length. It turns out tumor or cancer cells often shed shorter fragments due to differences in cell death relative to normal fragments. So just by looking at the length, you can increase the likelihood that that fragment actually came from a cancer cell versus a normal. And all of this is meaningless without the clinical data context. So really that's a very strong prior for us when we're entering a cell-free DNA analysis, thinking about where in that patient journey we're doing this work. Is it screening? Is it the diagnosis where the burden's high? Or is it monitoring where you need extremely low levels of sensitivity? So I'm going to tell 3 stories, all that focus on 3 different parts of that original curve I showed. So the first story is the CHUM Consortium, which Anne-Marie mentioned. Treating cancer early is the surest path to cure, but how do we actually find it? So this is led by the CHUM Consortium, our website's right there, and I've just highlighted in yellow the names of the graduate students and postdocs who led this work. This is actually a large national collaboration, started in Toronto, now involving 8 sites across the country. So the best way to find cancer early is to start with a focus on people who are at very high risk of cancer. So we focused on patients who actually describe themselves as ticking time bombs. These are hereditary cancer carriers. And the paradigm shift for us was not to look at any one hereditary cancer type, but really to look at that entire high-risk population as a single group. And that's really how we've approached this problem in CHARM. I've shown Angelina Jolie's photo here. She actually wrote a really compelling article for Time magazine, actually some years ago now, about her experience as a BRCA1 carrier and some of the prophylactic surgeries, breast and ovaries, having her breast and ovaries removed to really decrease her risk of cancer because of this wheel of uncertainty. So people who are hereditary cancer carriers often will enroll in provincial cancer screening programs, but these are often a single annual touch point with the healthcare system, asking the question, do I have cancer yet? If you don't, you sort of go back on this wheel of uncertainty. So you can't have more frequent testing, and often there isn't testing for the majority of cancers that these individuals are at risk for. And I've just shown one example from our a study of Li-Fraumeni syndrome with Raymond Kim and David Malkin, just showing this long tail of rare tumors that particular hereditary cancer syndrome confronts. And almost none of those have a clinical surveillance test currently. So our first entry point into this was in Li-Fraumeni syndrome, so P53 carriers. These are parents and their kids who have a pathogenic P53 variant. At that time, we actually deployed 3 technologies: a deep targeted sequencing panel, a shallow whole genome sequencing test, so we could look for copy number variants, and a methylation-based test invented actually by the lab next to mine, Daniel DeCavallo's group in Toronto, which is an antibody against 5-methylcytosine, so it's a very cheap methylation-based test. And you can think of these as sort of different needles that are in the haystack. We use 3 different technologies to pull out 3 different sizes and shapes of needles or cell-free DNA fragments. This was the first finding. So the way to read this plot, genome scientists love these kind of plots. Each row is a type of variation. Each column is a patient. All of our patients have P53 germline variants. That was the population we're interested in. And then we had 2 cohorts. We had patients who had Li-Fraumeni syndrome with active cancer. So in LFS, they get whole body MRI. So very comprehensive imaging test, and if a cancer was found, we turned them active cancer. Then we had LFS cancer-free, patients who had a whole body MRI and they didn't find anything. And the first finding was we found expected mutations. We found P53, a large number of P53 mutations, as well as a smattering of others. Kind of surprising to me was this blank spot here. I thought for sure we'd find P53 mutations in absolutely everybody, but we didn't. We only found them in 40%. So this is our first hint that gene panels were insufficient to find all the positive signal in this population. The other way more exciting finding were how many negatives were actually able to pick up by cell-free DNA. So, and half of those patients actually developed a cancer within the next year. And that gave us a hint that we were really onto something, especially for the negative patients, especially the ones who had cancers that maybe weren't being picked up by whole body MRI. And then the other relief was really how many of the negatives were truly negative. And we published that, I guess, last year. Since then, we've done the exact same study design in our hereditary breast ovarian cancer patients, so BRCA1 and 2. Exactly the same plot, the active cancer and cancer-free. The surveillance is a little bit different in this population. There's no test for ovarian cancer. In Ontario, we have the Ontario Breast Screening Programs, mammograms for patients with breast cancer. But really, the exact same story, but the percentages are a little bit lower. So we're down to 18% of the patients with cancer. We found a secondary mutation, so BRCA1, 2, P53, and PALB2. But also a percentage of the patients who are cancer-free still finding point mutations in their cell-free DNA as well. So taking what we learned from higher risk from any syndrome cohort, but really using the exact same strategy, have some success in the BRCA1 and 2 carriers. But both these studies taught us that point mutations were really not the entire story here. I mentioned the second test, we did whole genome sequencing. This is sort of a vignette from a single ovarian cancer patient, came in October 2018. This is the way to read this. The x-axis are chromosomes and the y-axis are copy number variants. So at this time point, clinically negative and genomically negative as well. Year and a half later, still clinically negative, but you can tell by eye how disordered this copy number track is. So there's evidence of disease using copy number analysis on this patient. And then a year and a half later, the patient came in off cycle because they were getting sick, they had symptoms, and then they were deemed clinically positive. But if they had access to this test as part of routine surveillance, this cancer could have been caught a year and a half previous. The challenge here is the copy number test is not that sensitive. So I showed you the best example. Most of the patients were actually negative. So this plot is blank for a reason. It's very unusual that we have a copy number positive specifically in our BRCA positive cohort. You can see the samples from that patient down here, maybe a few other hits here and there. But there's a real issue by using broad copy number profiling in isolation to find low levels of disease as part of surveillance. And this is where we turn to other features of the cancer genome. So if we take not just copy number variants, but specifically copy number variants that are supported by short fragments, so using just one other feature of cell-free DNA, you can see a lot more signal now pops out, because now we can say we're going to computationally enrich for the shorter fragments and then reconstruct those same copy number alterations as well. And this is really where we're starting to go as a lab, is starting to integrate all these different molecular features of cell-free DNA to boost the signal for copy number detection, mutation detection, and so on. The other piece of information, especially in the surveillance context, is not just to find cancer, but also try to figure out where in the body it is. And the workflow that we've been considering with the clinical geneticist has been find the cancer and then trigger a gene-appropriate or disease-appropriate clinical follow-up. So to do this, we're leveraging large open chromatin maps from the Cancer Genome Atlas. The way this works, cell-free DNA is not free-floating as long chunks of DNA. It actually floats and is protected in the genome by wrapping itself around these nucleosome proteins. And this is because the free-floating, the genome is like this. These open chromatin regions are not being protected, so they're actually being digested and they're actually depleted in regions of interest. So what we can do is take these open chromatin maps, look for decreases in coverage for every single cancer type, and use that to infer the type of cancer that we're seeing using those other assays. This analysis gives us maps that look like this. So I've put the nucleosomes across the top here, and you can see sort of these coverage waves that correspond with where these nucleosomes are positioned. And then we can ask a focused question for every single cancer type one by one. Is there depletion in coverage in the open chromatin regions that are unique for that cancer type? So here's a prostate cancer open chromatin reference. All of our LFS carriers and healthy controls basically show the exact same pattern, except for the only patient with prostate cancer. You can see they really have this depletion in coverage specifically across that large catalog of open chromatin regions. And same sort of trick with the bladder cancer patients. This is actually 3 samples from the same patient with bladder cancer, and they're the only patients that had the depletion in coverage. So no single region was actually informative, but it's when you aggregate tens of thousands of open chromatin sites where these signatures can be very strong. And similar story, just like in our BRCA cohort, if you look at the open chromatin regions for breast cancer, the patients who are positive for breast cancer show this depletion in coverage. Where we're working now is trying to figure out these patients, the patients who are cancer positive, but they don't actually have breast cancer. And you can see there is still some signal in those patients that is different from our healthy controls. And I think this is a problem with our reference set. So really trying to have a very high quality open chromatin map that is truly unique for each cancer type. And I think the only way we're going to get those high quality maps is through data sharing of cell-free DNA profiles from many patients who have any one of these cancers. You can see here, just like in mutations and copy number, picking up some of the signal even in the cancer-free patients. And actually it's the cancer patient, or the patients here with this depletion in coverage are the same ones that are positive for copy numbers, short fragments, and mutations. So you can really start to put all these together. So where are we going with CHUM? We've since been funded by the Canadian Cancer Society to turn this into a prospective study. Now 8 sites across the country, including here in McGill, or here in Montreal. We're enrolling 1,000 patients with hereditary cancer syndrome, and they can have any of these 5. So we have a lot of experience with BRCA1 and 2, Lynch syndrome and Li-Fraumeni. New are NF1 and CDH1. Patients are randomized to receive a clinical cell-free DNA report. So if you're in your experimental arm, you have a blood sample, we do cell-free DNA sequencing, and there's actually now a playbook of 30 different workflows that are all specific for patients depending on their clinical scenario. So if it's a BRCA carrier who's had their breast removed, the first thought isn't always breast cancer, it's ovarian cancer. So the clinical follow-up is kind of tailored to their clinical scenario. Patients who are in the control group, we still are collecting blood samples, and we bank those for future research and potentially as a validation cohort as well. And yeah, Anne-Marie mentioned we're ATOM users. All of our samples, and actually the reason this project has worked, all 8 sites are all entering all their samples into a centralized biobanking software. So we have eyes on, real-time eyes on how many samples are coming in and being tested at any one time. So since April 2024, we've enrolled 575 patients. Half of them have been randomized to receive cell-free DNA. 4 of them have been positives, and we've actually missed 1 invasive ductal carcinoma, so a little tiny tumor in one of the BRCA carriers actually was not picked up by this test. We intentionally have not analyzed the control arm, so we're going to wait until the end of the study to do as a proper randomized comparison. I did want to share just 2 vignettes on 2 of the positives. One of them is a BRCA carrier with multiple positive cell-free DNA blood tests. BRCA1 carrier had hysterectomy and her ovaries removed. A history of cancer, so that's where I put them into a sort of a different category. Breast, multiple ovarian cancers, and shallow whole genome sequencing, just like in the other ovarian cancer patients, showing multiple cytogenetic abnormalities. The subsequent test showed the exact same profile, so we felt like we were onto something. Raymond Kim was managing this patient, could not find the cancer with many targeted tests, so he bailed out with a PET CT from head to hip, and actually picked up a signal in her appendix, totally unexpected for a BRCA carrier, and she's actually subsequently had her appendix removed, and that tumor has been sent for whole genome sequencing through Marathon of Hope Cancer Centers Network. So that analysis is still ongoing, but really a nice chance where we could leverage 2 consortia, one interested in cell-free DNA and the other interested in cancer genome sequencing from really the earliest possible cancer. And the question there is this a return of the old cancer or a brand new primary? Unfortunately, we didn't have access to any of these older tumors to sequence. The second positive is a Lynch syndrome carrier, so PMS2. Similar story, had a history of cancer and of numerous interventions to decrease her risk. In this case, actually, it was a point mutation, or actually an insertion deletion, specifically for this patient, kind of like you'd expect from a mismatch repair deficient tumor. And the follow-up test found the exact same mutation, as well as a second mutation within the CDH1 gene. And not just any old mutation, it was also in a microsatellite site, so really reflecting the type of cancers that this patient would expect. Follow-up investigation actually found totally unexpected, what they termed as a metastatic tumor in her leg and in her hip, and then she was managed essentially as a patient with recurrence rather than as a new primary tumor. So 2 positives with 2 very different clinical managements from the exact same cell-free DNA result. So this sort of begs the second question, which is once a tumor is found, what are you going to do about it? So how do we know if this treatment is actually working? This is a collaboration through the Terry Fox Multiple Myeloma Molecular Monitoring Project led by Tony Riemann in the Maritimes, and also collaboration with the Mayo Clinic Multiple Myeloma Spore Project. And this is led by graduate student Dory Abelman. So really changing gears, this is not hereditary cancer at all. It's multiple myeloma. It's collected using this, I would consider a pretty invasive procedure. So this is a long bone marrow aspirate of the iliac crest and the hip, pulls out tumors, and then you use a magnetic bead to enrich specifically for cancer cells. Minimal residual disease has now been deemed a clinical endpoint by the FDA, and that's done using a targeted sequencing assay to look for a very unique IG rearrangement in the cells in the bone marrow. Big challenge with the bone marrow is patients don't want to undergo multiples of these, and the clinical trial we wanted to be involved in wanted to do testing every month. No one is going to sign up for a monthly bone marrow aspirate, so we wanted to investigate whether a blood draw would be a reasonable alternative. Around that time we were designing this study, this paper came out from Dan Landau's group that said, instead of looking at one mutation, which is what we've been doing in CHUM, why not look at every single mutation? So the idea here was, rather than sequencing on a gene panel to 10,000x, why not go way out here and have much greater sensitivity by sequencing 10,000 mutations or more across the entire genome? So don't try to get any one mutation perfect, but sprinkle your coverage around to have a chance to find enough mutations to have evidence for or against minimal residual disease. And the acronym here is kind of training your iPhone. It can be super targeted. You have one point on your fingerprint, or you can test the entire thumb. Even if the entire thumb isn't perfectly scanned, you can really predict who's who at very high accuracy. So the person's individual bone marrow genome is sort of that unique fingerprint. This is what the data looks like. So we have their bone marrow at diagnosis, so single bone marrow. We sequence the entire genome. In this patient, we detected 4,087 mutations. We also get some cytogenetics. We also found a point mutation. And then with that list from the bone marrow, we go looking for those mutations in the blood. So here's their blood at baseline, same time point as the biopsy. Even though the tumor had 4,000 mutations, we only found just over half of them. And that's okay. The goal is to measure disease. It's not to get any one mutation perfect. Patient underwent induction. They had a clinical complete response and also a molecular one. So the same number of mutations we found in their blood matched the same number of mutations in the healthy control. A year later, this patient's on lenalidomide maintenance. They had another bone marrow test at that point using the targeted assay and a PET assay, both negative. But here we already had a hint that this patient was going to relapse because we found 400 mutations above the background using the blood test. And then 6 months later, you can really see that signal starting to upswing, and that patient had actually quite a dramatic relapse. We could actually find a large fraction of their mutations in their blood. So that was sort of one example for how to read these plots. We've now done this in a much larger cohort. Just to focus on the first 2 plots at the top, this is the tumor-informed approach. So we had high-quality bone marrow from 31 patients. All the patients who relapsed all followed a very similar swing to that patient I showed on the first slide. High at baseline, went down during maintenance, and they have this upswing at the end. This is the patient I showed, this really dramatic one. And the patients who didn't relapse, they go down and they stay down, and they stay down really, really long term, some of these patients for years. The challenge is we only had high-quality bone marrow from 31 patients, but we actually had 36 patients. So 5 of those bone marrows were unsuitable for whole genome sequencing because the tumor content in that bone marrow aspirate was so low. So what we did instead is we took the mutations from the cell-free DNA at baseline. So we treated that baseline cell-free DNA. Since the tumor burden was so high, we treated it like a bone marrow aspirate, and we used that as the reference. And we actually found the exact same pattern, this down tick and then upswing for patients who are relapsing, and then non-relapsing going down. But I think the important translational point here is now patients who maybe had a suboptimal bone marrow biopsy could still benefit from MRD-based testing because they could be cell-free DNA informed. And that brings me to the last story. What happens at relapse? How do you help manage patients who are having relapse, especially relapse with potentially new mutations or new copy number variants that wouldn't be apparent from their baseline or their diagnostic sample? And this work is led by another graduate student, Bianca Kirsch, in a collaboration with the transplant group, UHN. This project also started out as a tissue project. This is actually Marathon of Hope project doing whole genome sequencing. Liver cancer actually had the record in my lab for the lowest success rate for genome sequencing. So literally half of the tumors that we were sent, we couldn't generate whole genome sequences, and it's because of this bridging therapy. So the protocol at our hospital, patients have HCC, but they're outside of the eligibility for transplant. They undergo this downstaging, so ablation, systemic testing, and radiation to attempt to shrink the tumor down. And the goal is to get these patients within the transplant eligibility, criteria. Then they go on the transplant list, and they still get bridging therapy. So that by the time those tumors get to us, which is after they have an explant, those tumors are highly necrotic, and we have very few viable cells left. So this is where we want to, of course, turn to cell-free DNA sequencing to see if we could pull that cell-free DNA out of the blood instead. Indeed, we could. So here's a single patient with their tumor, their blood plasma pre-surgery, and their blood plasma post-surgery. You can see these very nice copy number alterations, even better than the ones I showed you previously, nice focal copy number changes. If you look at their blood plasma pre-surgery, 8% is pretty high, so we could actually see those same copy number alterations there. And we could, just like in myeloma, we could find large numbers of mutations in their blood pre-surgery. But even post-surgery, I mentioned the copy number detection is not very good, but even post-surgery, we already knew this patient was going to relapse because we could find hundreds of mutations still circulating in their blood 3 months post-transplant. And actually, this patient did go on to relapse. But we actually had a hint that if we had access to this test, we would have had a hint that relapse was already known very soon after transplant. So the challenge here is, okay, we can find these mutations, but we can't find any of them with very high confidence. And this turns me to my last story, which is trying to reconstruct that cancer genome right out of the bloodstream by sequencing to really great depth, 200 to 400x coverage, pull out all the cancer-derived fragments, and use that to reconstruct the cancer genome. And that's returning to this model. So what we've done is we have a huge table of 60 columns and billions, maybe trillions of rows now, and we know which ones come from the tumor because they're all mutations. So we use that as labels. So we're able to label which fragments definitively come from the tumor because they have that somatic mutation. And then now we're able to use machine learning techniques to score all of these different features. Overlap with open chromatin regions, looking at short fragments, looking at methylation marks. Don't really look at the T-cell receptors. We could if we wanted to. And then trainees across a large numbers of patients. So we had the tumor genomes from Marathon of Hope, and then we're able to use other funding from CHR to sequence the whole genome from the plasma and essentially compare the two. So this is one of the first, I guess, success stories from this project. Still pretty early days as we pile up large numbers of reference tumors and cell-free DNAs. Here's the liver tumor here, very clear copy number tracks. Pre-surgery, you can see there's certainly signal even by copy number. The challenge here is post-surgery, all these raw plasma tests are all negative. You can't see anything. But then you can take that exact same data, upgrade the coverage, and then pull out only the cancer-derived fragments, and then run the exact same algorithm. And now you can see, you can actually predict really with high fidelity, this is almost 100% tumor fraction, the exact same copy number tracks that you can see in the liver tumor. I say this was a work in progress. The eagle-eyed people in the audience might notice that we've missed all the deletions. So you can see all these deletions here. we're very bad at classifying the absence of disease. So we can use the mutations and things that are there, where we're still struggling is trying to tune the weights on all of the other features to try to pick up a lot of the non-mutant fragments, and especially the deletions, because we have less data circulating in the blood because those tumor cells don't have those fragments. But the idea here is really to put all this data together into one place so we can make these models better and better. And the goal here is, if you can have increased depth and you can reconstruct that genome at relapse, this actually opens a whole new type of clinical trial that could be based specifically on the genome inferred from the cell-free DNA. And this is a project we're working on now. We've got about 10 groups from all around the world interested in deploying some of this infrastructure we set up for whole genome sequencing. The idea is to do unbiased genome sequencing across the patient journey, share all that data in a centralized database, and then do basically leverage all the machine learning algorithms that are now available in the cloud to look for some of these patterns to let you differentiate a cancer cell from a non-cancer cell. And really think of this as pan-cancer, especially important for the patients who are in CHUM, who are at risk for so many different cancer types. And this is really my last slide. This is where we want to go. Really thinking of cell-free DNA sequencing as a singular approach that you can tailor computationally to different points in the patient journey. A blood checkup with your frequency tailored for your risk profile. That's some of the work we've done in CHUM. Try to find these cancers early, so not just at lower burden, but also earlier in the time course. Make MRD testing routine specifically from the blood and train these models specifically. We're starting with myeloma. We want to broaden this to other cancer types. But same sort of story, try to capture some of these relapses earlier while burden is lower. So really have these highly sensitive techniques, but really using this whole genome sequencing test as a singular platform. So yeah, thanks to all my many collaborators from across the country. Patient partners are actually really important for this. They're the ones who encouraged us to go for the randomized controlled study, which is quite unusual in the cell-free DNA world. This is something that they specifically encouraged us to pursue. If you know anyone interested in participating in CHUM, the other trick to CHUM I forgot to mention is making the patient experience very seamless. So we actually don't ask the patients to come to the hospital. We have a deal with Life Labs and DynaCare. They can just go to their local blood lab to provide their blood every 4 months. So really trying to make this research sort of part of the regular surveillance and allow that testing to happen more frequently. And then, yeah, certainly having access to whole genome sequencing through Marathon of Hope has really been a critical accelerant for a lot of our work in cell-free DNA sequencing. Even though that network isn't all about cell-free DNA testing, all those tissue genomes have been a really valuable data resource. So thank you very much, and happy to discuss further in the panel.
So as our panelists come up, I'd like to introduce the person who will be moderating this, who is Isabelle Michaud from Boehringer Ingelheim, who's the patient access manager for the province of Quebec. So Isabelle has over 25 years of experience in the pharmaceutical industry with expertise in market access, health technology assessment, and stakeholder relations. She's a graduate from the Master's Program of Health Technology Assessment from the School of Public Health at the Université de Montréal, l'ESPOM, and she is recognized for a strategic patient-centered approach. So I'd like to invite her up to start this.
Bonjour à toutes et à tous. Boehringer Ingelheim est fier de s'associer au CRCHUM pour la conférence Prestige. So thank you for joining us as we explore a topic of the intersection of cutting-edge research and clinical practice, the implementation of liquid biopsy in research centers and hospitals. We've just heard an inspiring presentation by Dr. Pugh, who demonstrated how whole genome sequencing can transform cancer detection, diagnostic, and monitoring. We'll now take the conversation further with a panel of experts whose work is helping shape the future of precision oncology in Quebec and across Canada. So joining Dr. Pugh on the panel are Dr. Burnier and Dr. Antoine Désilet. So Dr. Julia Burnier is an associate professor in the Gerald Bronfman Department of Oncology and the Department of Pathology at McGill University, and a scientist at the Research Institute of the McGill University Health Center. She leads the liquid biopsy lab within the Cancer Research Program and directs the circulating biomarkers of Health and Disease Network. Her research focuses on tumor-derived circulating molecules to identify biomarkers and therapeutic targets with the goal of improving cancer detection and monitoring. Dr. Antoine Désilet is well known here at the CHUM, so he's an hematologist oncologist at the CHUM and a researcher at the CRCHUM. After completing his medical training at McGill University and subspecialty training in medical oncology and hematology, he pursued a research fellowship at Memorial Sloan Kettering Cancer Center in New York. His work focuses on molecular diagnostic, early phase precision oncology trials, and the use of liquid biopsy in thoracic and head and neck cancers. He also co-directs the Guy Lafleur Precision Oncology Program. So thank you for joining us. So as Dr. Pugh highlighted, liquid biopsy is emerging as a powerful complement to traditional tissue biopsy in oncology. By analyzing circulating tumor DNA, circulating tumor cells, and other blood-based biomarker, it's enabled the detection and actionable mutation to guide targeted therapies, monitor treatment response, and identify resistance earlier. In my role at Boehringer, I've had the opportunity to engage with patient communities through various patient associations. And it's clear that there's a growing interest by patients in seeing liquid biopsy integrated more fully into oncology care path. This growing interest reinforces the importance of understanding how liquid biopsy is currently being applied across Canadian institutions, what's working, what challenge remains, and where the greatest opportunity lie to advance its role in oncology care. To start the conversation, I'd like to ask our panelists the following question, and maybe Dr. Burnier, you'll be the first, if you want. How is liquid biopsy currently being integrated into clinical practice at your institution, and to what extent has it become part of the standard of care in oncology?
That's a great question. The decisions for how liquid biopsy is being integrated from a reimbursed point of view are, of course, decisions made at the provincial level. So in Quebec, we have one test that is now being reimbursed within the lung cancer space, and we have in the pipeline several other tests that we think will be, let's say, coming out in the next 1 to 3 years. Across Canada, that looks differently from province to province, but I think ultimately the approach has been similar in how we get things into the clinic reimbursed. That's one aspect of having access to liquid biopsy tests. The other one is through clinical research studies, through clinical trials. And I think that in Quebec, as well as I'm certain in Ontario, is a very big area of not only research, but actually applying different ctDNA assays that can be used within the clinical setting and can inform treatment decisions. There's many ctDNA-guided clinical trials ongoing in different types of diseases. So I think that there's a lot of movement in the area. But when we talk about true standard of care, it has to be linked to a clinical need and an actionability of those results, like anything else, like immunohistochemistry, like pathology. So I think we have to ensure that the research that we're doing now, whether it's in the clinical space or in the more basic space, is really going to be the next generation of assays that will meet specific clinical needs, where we have therapies that can be given to patients based on the results, or actionability that can change based on those ctDNA assays.
Yeah, I could add the Ontario context, which was very similar. Certainly lung cancer has led the way there. I think the real opportunity is dissolving that wall between research and clinical care and really making sure that the environment is ready to pick up some of these technologies, especially in liquid biopsy. It's incredible how quickly those technologies are accelerating. Commercialization has been very successful in this area. Huge opportunities for machine learning and AI to make those tests even better. But I think that missing piece is that translational layer Julia talked about between basic research and full-blown clinical standard of care. And clinical trials have kind of been that incubator for a long time, which I think has worked very well. It's a very controlled environment. But I think building some of the infrastructure to try out those assays and industrialize academic software and approaches to be clinical grade and then make that jump in that case to standard of care. I think ensuring that there's a clear pathway from basic research all the way through to standard of care, but then back again, making sure that patients can participate in research and feed back some of that data that's going to make the next test better and better. I think that's a missing piece, I think, in a lot of the right provinces. I'd like to see maybe more of that little translational piece between basic science and clinical care.
Maybe adding about the current context in the province of Quebec, so we discussed what's currently available in a clinical setting. So considering the actionability of the results, most of the access we have that is currently refunded is in the context of non-small cell lung cancer, more specifically in the context of advanced cancer. So considering patients at diagnosis, we have 4 specific indications. And this is basically the framework for all of our indications. So we have patients who have advanced disease that is not deemed to be easily accessible for biopsy testing for tumor NGS, patients with inadequate samples also for the initial tumor NGS, patients with a disease trajectory and aggressiveness that do require a very rapid test result in less than 10 days, and also patients following progression on targeted therapy approaches for whom second-line therapy options are available based on those resistance patterns. So for the moment, this is the refunding we have, and the public discussion concerning, liquid biopsy really is new in the province. We have a positive recommendation since August of 2024, and the formal refunding was issued as of a couple of months ago, so as of April. So currently, there are 2 major hospitals in the province which are joining most of the liquid biopsy testing in the clinical setting, being the McGill network and also the CRCHUM in Quebec City. And those are based on different assays that are also getting validated through time. So most of the testing is centralized in specific centers. They're being sent from the CHUM at the moment to those 2 specialized hospitals. And the goal is just to increase the access for those trials. And this is really following clinical practice and how actionable those results can be. So we have a strong recommendation for lung cancer, but more and more as clinical trials are published, we do see that there's actionability even outside of the context of lung cancer.
Thank you. So it's not yet fully integrated in some centers. What do you think are the main barriers besides funding, but are there other barriers?
Yeah, I think what Julia and Antoine both talked about, it's the clinical, what do you do with it question. Like, what is that pathway? And I think that's why CHUM took a year to get started, because everybody who was charged with managing the patients didn't want to get stuck with a ctDNA result that they couldn't do anything with or didn't know what to do with in the moment. And that's what led to that playbook of all these different clinical follow-up tests, because that work had to go in to have everyone agree what has to happen when you get that test. It's no help to just throw a pot into the lab, to throw a result over the fence and hope something's going to happen with it. So I think that's actually probably the #1 barrier. Policies and guidelines and standards, probably more on the clinical side for what you do once you actually get a cell-free DNA test.
And I'll add that, we're in an era where patients are really involved in research and really involved in the work that we do. They partner with us, as Trevor has shown in his work as well. So it's challenging to enroll patients onto clinical studies that are research only, with them knowing about liquid biopsy and that there is a test result available, and what to do with that information. So I think managing how we report what is actionable, not just in terms of there is a therapy out there, but what do we have access to in Canada as well? In particular cancers, you know, breast cancer comes to mind with ESR1 mutations, and, you know, there's actionability in certain parts of the world for ctDNA assays where if you don't have access to the drug here, is there a reason to be doing this test or to providing even additional stress onto patients? So I think there's a lot of research that has to be done, both from the ctDNA liquid biopsy biology aspect, but really also from an implementation science perspective of how do we implement this within our current funding model, how do we engage with patients to know what type of results they would like to see and how, the format of that. So I think there's a lot of space for us to learn. Liquid biopsy is not the first sort of companion diagnostic to come out there. There's many, you know, I always go back to pathology, immunohistochemistry. So we have ways to, you know, precedents to think back to and to guide us. But I think that there's still a little bit of work to be done between bridging what we believe to be true within the research space and what is informative, helpful, and practice changing on the clinical front.
Totally. So I think the first aspect really is the actionability of the results again. So what do we do with the results? And then in many instances, including in the context of genotyping for liquid biopsy, you'll end up with a possible biomarker, and then the clinician just ends up scratching his head and thinking, what are the next steps? Are there any possible targeted therapy approaches, any clinical trial matching? So the whole infrastructure really needs to be built, I would say, at different time points simultaneously. So first of all, you develop the test, you also validate it, but you also educate the clinicians, the patients, and the general healthcare population on how to manage those results. And then the goal is also to basically match those results for possible targeted therapy approaches if they're, reimbursed, or in the context of clinical trials, special access programs also. So all of those needs to be kind of put in place at the same time. Additional issues really are access to sequencers also, so it seems like a no-brainer, but all of this is pretty much focused in specific centers, as I alluded to earlier on. So we're currently dealing with a high volumetry in terms of tumor NGS at the CHUM, McGill Hospital, Quebec, and Sherbrooke, also the main academic centers. And we're already kind of saturated in many instances. So how can we go on and develop additional assays? This is also a big challenge in a clinical setting. There's also assay-specific questions. So for many people, liquid biopsy is kind of a very generic term, but as we know, and as we'll also discuss, there are multiple possible applications, and most of what has been developed and accessible has been in the context of genotyping. So mostly tumor-agnostic assays that are quantifying different variants as part of limited panels, and most of the implications are in the context of specific therapies for actionable biomarkers. So biomarker-derived drug or clinical trials. But there's multiple different applications, including in the context of quantitative ctDNA analysis for surveillance of cancer. So what is the lead time between actual ctDNA testing and radiological progression? So we know that from convergence literature that ctDNA is way more sensitive than the radiological results we normally rely on in oncology, considering the limited resolution of those imaging. So this is still an unmet need in the clinical setting, and even in the context of MRD testing, so minimal residual disease testing. Most of what we use in the diagnostic setting really is limited. So we're measuring disease radiologically speaking, we're using physical exam, but all of those parameters really aren't focused and patient-specific. So most of the work that has to be done, I would say, is outside of the genotyping space also, because there's currently very limited, if no even FDA-approved assays in the context of MRD or quantitative testing. And then increasing also the access to genotyping and the qualitative aspect of liquid biopsies outside of the non-small cell lung cancer population.
Yeah, I'd like to pick up on your point of the infrastructure. I mean, not just the sequencing bottleneck, which it definitely is real, also the computational part. As we've moved from panel whole genome sequencing, we crippled our institutional high-performance computing cluster, and we're now in the midst of moving everything to the cloud because we just couldn't achieve, we weren't able to scale fast enough relying on traditional infrastructure grants, which take a year or two. You can't wait that long when you need access to compute and storage immediately. So I think having especially a strategy across provinces for how do you scale these very large genomic assays, but then have the endpoint still be a clinical endpoint. So burst to the cloud, make use of the amazing packages and technologies for mining that data, but then still cook it down to a report that you can actually pick up. So I think there's actually two bottlenecks in the system that need some investment.
And to confound that further, as AI and machine learning tools get more convoluted, that's going to accelerate that problem or that issue further, or that challenge. So I think that coming up with ways to manage that as big data is made available, to have computational power to manage that data.
Yeah, and making sure our data are structured to be AI ready. You can't put it in a big Excel sheet. There are ways to structure and store your data in ways where you can try out a bunch of algorithms in a very proficient way. And not just the genomic data, but the clinical data as well.
I think we have time for one more question. All right. But should we open for a question right now, or? Okay. So one last question. So while liquid biopsy is more often discussed in the context of oncology, there is a growing interest in its potential application for other disease and conditions. From your perspective, what are the most promising non-cancer use in liquid biopsy?
Yeah, Julia, I spent almost an hour today talking about this exact topic. I don't know if this is planted or what, but yeah, there's huge opportunity. The cell-free DNA, most of that signal is actually not cancer. It's all the other cells in your body. So big opportunities to look at immune responses, immune health. We use that open chromatin algorithm to look at cancer signal, but you can also look at T-cell signal and B-cell signal. So really, I think the sky is the limit. We've just had one conversation locally in the lab. Currently, we do germline testing based on cells in the blood, but we kicked around the idea, well, what if you did cell-free DNA testing? You could still get the germline hit, but then get all these other interesting cell-free DNA biology at the same time. So yeah, I think actually the field is very broad for where it could go, but as a geneticist, I sort of thought hereditary genetics is kind of a good, easy place to start.
And I think going back to the idea of big data, I think we have to stop thinking of patients as being defined by one particular disease or health state. So, we often think about the cancer patient as, a cancer patient, or the person who has a transplant as a transplant patient. As Trevor has already mentioned in his talk, we borrowed the concept of ctDNA from the NIPT, the non-invasive prenatal test. So this distinction of fetal DNA from maternal DNA is really the same concept we use to distinguish cancer DNA from non-cancer DNA. So as we start to think of people as more complex humans, we can think that a woman who is pregnant today could develop cancer tomorrow. A person who has cancer today could receive a transplant tomorrow. And so by having access to, let's say, their cell-free DNA secretome, or the genomics of of all the cells in their body are secreting, I think we have an opportunity to be able to use different pattern recognition tools to see if we could predict, for example, cardiovascular outcomes of chemotherapy. This is a big issue. It's not my area of expertise. I'm throwing out different things. Infectious disease. So a part of what we've done at McGill with our networks is exactly that. Can we transcend different disciplines and create a cell-free DNA network across diseases? So we have a study in tuberculosis for cell-free DNA. We're looking at how exercise and lifestyle impacts basal cell-free DNA levels, which could impact later when you have a ctDNA assay and you have, for example, very high levels of normal or non-cancer cell-free DNA being secreted. So I think there's a lot of questions, and I think moving away from, defining people by specific illnesses at a particular time, we can learn a lot about how cell-free DNA can inform more generally about health states.
Totally, and we always allude to oncology practice, and we're dealing with very, very small tumor fractions and scratching our head in terms of sensitivity testing and LOD for many of those assays. But in many clinical contexts, including for inpatients, We have patients with solid transplant, which are dealing also with possible reject of their graft. We can monitor also donor-derived DNA, those populations, to see if there's any evidence of reject, even on a real-time basis. For patients in the infectious disease setting also, including immunocompromised patients with atypical microorganisms. We're often very much, limited by cultures and by the rapidity of access to those diagnostic procedures. And again, in such setting, DNA and liquid biopsy testing can be a huge plus to diagnose those possible atypical organisms, but also diagnosing nascent resistance also patterns for stewardship and antibiotic also use. So the applications are extremely broad. We have to think about liquid biopsy as more of a diagnostic tool than specifically in the context of cancer. In many of those instances, so oncology, infectious disease, even solid organ transplant, one of the main issues will be to address the turnaround times also, because it's not easy, but it's more convenient to batch all of those patients, retrospectively also analyze those cases. But you want to deal with an actionable result, it needs to be present and it needs to be also available fast in many patients. So accelerating also, and then there's always a cost effectiveness in terms of how many samples can you batch together, the volumetry, the samples, the reactants. So it really is a tough question, especially so in the context of a public healthcare system. So I think most of our work would also be in the clinical setting, how fast we can obtain those results and how we can implement changes in our practice also.
Thank you. We now have time for questions, so please feel free to come to the mic.
I'll start. Well, thanks everyone for the great discussion. Just considering potential actions that we can take from these analyses. So we're entering an era, or re-entering an era of cancer vaccines, precision medicines, specifically on cancer vaccines. So how can we accelerate the development of personalized cancer vaccine based on liquid biopsies and circulating DNA?
Yeah, I guess I could start. That's been a major motivation for that genome reconstruction activity for the liver cancer project, was reconstructing the genome to find new antigens, but also use the fragmentation patterns to infer expression of perhaps non-mutant neoantigens as well. So there's actually two strategies at play where that cell-free DNA piece is quite important. The third part we haven't done, but we really want to, is infer immune response from the fragmentomic profiling as well. So post-vaccine, you can make the statement there has been immune activation or not. So I think it actually has a role for personalized or potentially generalized design of vaccines, but also monitoring those vaccines over time. Yeah, this is a huge opportunity, especially for Canada. We've got great local expertise to do this, and as other countries are not investing in it, this is our chance to really jump in, and all that cell-free DNA data can be reused for exactly that purpose.
So at the moment, can we generate vaccines based uniquely on the liquid biopsy or not?
I would probably argue no, but I have a grant with David Malkin and Nuri Tabori to do exactly that. So we think that future is there, and we had funders put their money where our mouth is. So I actually think that's actually quite a realistic question. I think the big open question is, do the vaccines have to be personalized or generalized? And I think that's where agnostic genome approaches are very important, because it's by definition personalized. So if I had to choose between the two, probably personalized is going to be the way to go. But yeah, that's the main question in that grant that we're working on.
And maybe it's not going to be personalized for everybody, but depending on the type of disease as well, because we see commonalities in certain cancer types that are less readily seen in other cancer types that have more heterogeneity between patients. So there might be an opportunity to really look at both. I think in terms of, detection of possible targets or candidates for vaccines, I think there's a lot in the blood, and not just the blood, but, we can talk about saliva, urine, other biofluids as well. We've talked a lot about cell-free DNA, but, the other love of my life, extracellular vesicles, are also very important because in a way they stabilize the DNA and you don't have as much degradation. So there's another opportunity to see what are in these nanovesicles and that you can find from a liquid biopsy as easily as any other analyte. So I think there's going to be a lot in the next 5 years in this space.
Yeah, as you know, in the clinical trials, this design setting, there's huge interest in terms of the adjuvant setting for non-small cell lung cancer and melanoma patients also, and personalized neoadjuvant therapy, which is what we use in terms of cancer vaccines and nomenclature. So this is currently relying on tumor, identification of specific neoantigens, which are then translated through RNA vaccine infrastructure into personalized vaccines. I think one of the dreams we could have also in the near future would be to perform more non-invasive genotyping on specific ctDNA from liquid biopsies and maybe adjust those cancer vaccines throughout the patient's journey. So based on possible associated dynamics in terms of cancer growth, but then reproducing specific, vaccines that are based on the change in terms of proteomics also in the patient to the trajectory. So again, turnaround time issues and also sequencing costs can be prohibitive, especially for whole genome sequencing and deeper also parameters, but hopefully we could get there in the near future.
Great, thanks everyone. Regarding costs, because you opened that door, what are we looking at in the next couple of years? Twofold, tenfold, 100-fold decrease in cost of sequencing?
Yeah, it's dropped 50% in the last year. There's been a lot of new competition in the sequencing space. Very interesting to see how the market dynamics are playing out there. Genomics has always gotten cheaper year over year. I don't know about 10 times, but I could imagine it like in the next 5 years. It's becoming very commoditized. Actually, our big cost driver is not as much the sequencing, it's the people. The people in the lab do the capture, and it's actually why we don't do as much targeted sequencing, because there's so much labor to do that capture. It's actually cheaper to just put it on the machine. So I think sequencing reagents are going to get cheap. I think the infrastructure issue is probably the bigger, because that's a big fixed upfront cost that's going to need. That's not getting cheaper. That's going to need some investment. But the little snippet that's sequencing, I'm quite confident is just going to keep getting lower and lower.
Hello. You mentioned earlier something about making sure the data is AI ready. So I was wondering in terms of, could you be maybe more specific about the criteria that by which one would judge whether the data is AI ready or not?
Yeah, for us it's been data standards have been very important. Documenting how the data was made. There's sort of famous examples where you have, from the imaging world, the machine's very good at learning the logo that's burned into the images. So like making sure that there's not funny little confounders that you don't necessarily know about. Structuring the data is part of it, but there's good algorithms to clean those up. But really making sure that our approach has been fragment level annotations, making sure we have consistent annotations done in the same way on every single fragment. And then they've been put into a very performant database. We use 4J graph databases to make sure that we can traverse all of our data very quickly. So there's an aspect of upfront pre-analytical standardization, there's structuring your data, but also modeling your data in such a way that it can be traversed very quickly, especially as it gets very large.
Small question. Since you have liquid biopsy data from before the patient get cancer for a patient at risk, and also after, could you do some, is it realistic to think to do some predictive analysis to find maybe earlier biomarkers of cancer that could help identify it even before? Or is it something less realistic?
I think you're referring to like screening for cancer?
Yes.
So there are some commercial assays already available. If you've heard the news out of the NHS in England, they've put in quite a lot of money into the Gallery test to do a population screen for cancer. I think it's really an exciting field. I would say that right now where we are with technology, I think our resources are better utilized for either at-risk populations, which was the talk that we've heard about, or in monitoring patients in terms of MRD, minimal residual disease, or treatment response. I think that's where we are clinically ready. I think that that said, I think it's very exciting to think about, you know, a possibility of having this as a screening modality. But again, I think in Canada, at least with the technologies that we have, if we can try to concentrate on those that we think could be at risk. You know, one area that we're working a lot in my group is on HPV ctDNA screening, both in oropharyngeal cancer, but also in cervical cancer. And for cervical cancer, it's not the need necessarily because we have pap smears, but for example, accessibility to pap smears. So we know that our indigenous populations in the north of Quebec and Ontario have lower access to screening modalities. There's higher rates of cervical cancer, worse outcomes. So could cell-free DNA, which could be collected, for example, through a vaginal self-swab in the comfort of somebody's home and sent to the lab, could that be one way for us to screen for a cancer in a different way and improve outcomes in specific remote areas that have, you know, disparities in access to screening tools. So I think we have to, it's not widespread yet. I think we need to hone in a little bit on specific populations, and I think the hereditary cancer population is a fantastic one. But I think we're not far, you know, to seeing this become more widespread.
Yeah, I think it's, as Julia was alluding to, it's a question of cost, scalability, and also access in such cases. So we know that in many cases, we're dealing with the limited sensitivity of liquid biopsy testing, and as we try to improve and to build also on our assays through computational also error correction, sensitivity can improve, but then the trade-off is also some possible false positive, and especially in the clinical setting, how do you deal with false positives in the context of patients that are undergoing screening without radiological correlation? So I mean, in the context of limited resources, the cost also of testing, hopefully we'll get there in the future or as assays are improving. But by definition, as those are, I mean, uninformed tumor agnostic assays in the context of screening, we're very much limited by sensitivity and also how specific we are to specific tumor sites and cancer histologies. So hopefully getting there soon, but I think it's a question of maybe doing screening procedures as part of patients with a higher pretest probability, including smokers for lung cancer screening, or still limiting our resources to at-risk patient populations.
Thank you very much.
We all know we have a transportation strike, and so we're mindful of the time. I'd like to take this opportunity to thank Boehringer Ingelheim for having sponsored this really exciting discussion, and Isabelle, Michaud for having moderated this part. I want to thank Trevor Pugh for his presentation, and all three of you, Trevor, Julia, and Antoine, for a really stimulating discussion. And I invite you all who've signed up to join us for the cocktail afterwards. It's an occasion to really get to know the different speakers, but also ask any questions you were too shy to ask in front of everybody else. Thank you.